Right. Our developing collaboration between the Smithsonian Institution and San Francisco State and the Tiburon Center, I think it's very exciting, and we'll talk a little bit about that momentarily. So I'd like to start, though, by stepping back a little bit, way back, out into the universe, to start where we are and show you, just remind you how unique of a place this is. Not Tiburon, which is also very unique, but planet Earth in general. So if we were some thousands of light years out in space, this is what it would look like if we were looking at planet Earth. You all know this from seeing the original Cosmos, not the one now, but the old Carl Sagan one. And as we get closer, this is what we see. And it so far is something unique in the known universe, which is this blue planet. This is, of course, the view from the moon that first came out in the late 1960s when astronauts first walked on the moon. And this was an incredible image, I think, that just caught everyone's breath, to really see how unique and isolated, really, the Earth looks in space. And it gives us a sense. Also, I think I can say of how precious it is, and that's really why we're here today. So it looks blue. This is the thing, the blue planet. This is what is the most striking feature of the Earth from space, and that's because of this very, very thin film of blue water on the surface of the ocean. But of course, as we come closer, and from our vantage point, where we stand on the surface of the Earth, it's not just a film. It's a swirling, three-dimensional, amazing ecosystem of all kinds of marine organisms. This is obviously a coral reef. We don't have those in San Francisco Bay, but there are equally amazing systems all over the place and different kinds of habitats. And I was just told that dolphins have come back to San Francisco Bay for the first time a few years ago in a long time. So what a fantastic success story. I fell in love with the ocean long ago as a result of these amazing organisms, and decided that that's what I wanted to do for my life. I couldn't actually believe that you could get paid to go to these places. But of course, the picture is darkened somewhat as we've come through time and found out how threatened so many of these systems are. But let me first give you an introduction to this concept that I'll talk about throughout the talk of biological diversity. So this is a sort of a science geek term for the variety of life, simply put. And there are different ways of thinking about it. We can think about it in a so-called taxonomic way, that is the way you classify life. There is a variety of genes within populations, as you can tell from looking around you. The genetic constitution of one species, our species. These systems have many. We can think of diversity in terms of the number of species present, families, phyla, and so on as we go up the taxonomic hierarchy. But that's basically talking about who is there. And how many there are. But we can also think of diversity in a functional way, that is, in terms of what all of these animals and plants are doing. And this is, I think, really the frontier for modern biodiversity science, and that's what I'd like to talk about today. So, for example, we know that ecosystems, or these large systems like coral reefs with all of the organisms in their environment, consist of species, but some of those species are doing similar things. They can be called functional groups. Those are all together in a community of fishes. Some eat plankton, some eat algae on the bottom, and so on. So there are various levels of classifying biological diversity. We don't need to get too much into the details now, but the important point is this ecological or functional aspect to it. So again, the challenge is that these beautiful natural systems that we all know and love are very much threatened now by a variety of different pressures and challenges, overfishing, nutrient pollution, invasive species, something that you know a lot about here in San Francisco Bay. And so one of the really pressing issues in biodiversity science nowadays is not simply describing who's there, but finding out what they're doing, why they're important to us. And so, in other words, there are many ways that there are many reasons why biodiversity or natural diversity is important, aesthetic reasons, even spiritual reasons. But one of the most important that we're thinking of nowadays is the functional consequences that loss of biodiversity has for what we depend on nature for, the so-called services or the production and stability of shorelines and so on that we rely on in nature. So the functional consequences. So just to give you a few examples of the importance of what I might call living infrastructure or natural infrastructure, we got a little bit of a rude awakening to this with Hurricane Katrina in 2005, when we obviously had a very large hurricane that inundated a large part of the city. There was a tremendous misery and loss as a result of that. There are many reasons for this. There's some indication that it might be the changing climate. We know that the climate is changing. Whether any individual event is because of that or not is a little bit uncertain. But we can be reasonably certain that more of this rising sea level is on the way. And part of the reason for that has been the loss of this natural infrastructure. So I was amazed to find out that over the course of the last 70 years, the state of Louisiana lost an area the size of Delaware, most of it in wetlands or salt marshes. And the reason that's important, of course, is because this is the coastal buffer that protects New Orleans and Louisiana from the bad weather coming in off of the Gulf. So this is an example. We hear a lot from time to time about the crumbling infrastructure that we have and bridges that need to be repaired and so on. But there's also natural infrastructure that we depend on, and this is one of the examples. In fact, there have been some quantitative studies of the economic damage that has been caused by hurricanes in the United States and how that was ameliorated by wetlands. And in fact, this one study that was done on something like 30 hurricanes in the United States found that the economic damage from hurricanes was lowest in areas with abundant wetlands, as we would expect. And in fact, estimated the value of that wetland protection against storms as $23 billion a year in the United States. So these are significant numbers to talk about. Protecting the coastline is not the only natural infrastructure that we have. A large part of our protein comes from fisheries. This is a video that came from the Millennium Ecosystem several years ago that shows basically the wave of fishing fleets going throughout the ocean over the course of the last 50 years or so. So it begins in the 1950s, and the red that we're seeing is the area of highest catch that has occurred during history. You can see that it's always on the leading edge of where these fleets are moving, and behind them is reduced catch. Between 1950s and approximately 2000, the entire ocean had been covered. That doesn't mean we're not catching fish there anymore. We are. Some of these areas are very productive. But what it does mean is that the largest fish and the highest catch rates were almost invariably immediately when they started fishing there. So there's a big effect of humans on the fishes, not just close by and in the Chesapeake Bay, but throughout the world oceans in remote places. Interestingly, there are aspects of biodiversity that appear to stabilize the fish catch for us. So this is from an analysis that a number of colleagues and I did some years ago, where we basically looked at the fishery production for each of the world's 64 large marine ecosystems. So each of these colored areas is a place that's considered, for some purposes, a large marine ecosystem. There are data on fish catches from those areas, and we also have data on the number of species of fishes that live in those areas. And what we find is if you look across the world at these patterns, that the average catch of fishes increases as the number of species in that system increases. So in other words, where you have this high diversity, a diversified portfolio, if you will, of fish stocks, you have a high return rate. And what's even more important in some ways is that in areas where you have abundant fish species. Richness simply means the number of species there. This is on a log scale, so 100, 1,000, 10,000 fish species. Where you have more fish species, you have fewer collapsed taxa. That is, fewer occasions when the fishery out-harvests the species and there's nothing left. So in other words, the diversity of these fishes provides food security in these areas. The reason that this happens is that the ecosystem, including the natural diversity that lives there of organisms, plants and animals, has a complex network sort of structure that is able to adapt to changes. So we can think of thinking of a seagrass bed, for example, which is one of the types of habitats that I work on, and which you have here in Chesapeake, or sorry, the other bay, San Francisco Bay. I won't let that happen again. In the seagrass beds, we have these food webs that consist of seagrass, obviously, and algae, and various kinds of small bugs that eat them, small invertebrates, and then obviously on top, we have the things that we like to eat. And I should put a human up on top of here because, of course, we have a big impact on the crabs and the striped bass and other fishes. But we can think of that web of interacting species as a sort of natural infrastructure that is producing products that are useful and valuable to us. Natural services or ecosystem services, and I've already mentioned a few of these. Sustainable seafood is obviously one of these. This is happening for the most part. Wild capture fisheries happen with relatively little management, in some places more than others, but out on the high seas, for example, we don't farm them, we don't fertilize them, and so on. This just comes from the natural ecosystem. In addition to the seafood, these natural systems clarify water, stabilize the shorelines, and so on. So the question that has really arisen to be sort of a frontier question in marine science and in science generally over recent years is asking about how these two things are connected. How does the change in biodiversity that we're seeing, the loss of species that we're seeing as natural habitats are converted, how does that relate to the resilience and the ability of ecosystems to continue to provide these services and products that we need and in a stable fashion? So this is the question that I've been interested in for some years, and that I believe we're beginning to tackle with this collaboration that we're beginning here at the Smithsonian with San Francisco State and others. So how do we get at this question? It's a big, bold problem. I should say it's a big problem, a big challenge that requires a bold, ambitious program. And so the Smithsonian, before I arrived 6 months ago, had been planning and thinking about how to do this for some time. And part of the approach for understanding how biodiversity influences resilience in marine systems comes from the model of what was done on land. And this is from what began as the Smithsonian's Center for Tropical Forest Studies. It's now known as Smithsonian's Forest GEO, Global Earth Observatory. And basically this started with a couple of Smithsonian scientists about 30 years ago puzzling over the question of why are tropical forests so diverse. So you've all seen the nature programs, perhaps you've been to some of the tropical environments and seen some of these tropical rainforests that are really sort of an archetype for biological diversity, lots of thousands of species of trees, all kinds of interesting animals living there, and so on. Why is this? Why are they so much more diverse than they are in Canada, for example, where you've got a few species of trees? So this is a deceptively simple question, but it spawned a huge research effort that was coordinated by the Smithsonian and has gone on for over 30 years, ultimately resulting in something like 50 plots. And these plots are modeled on the first one that happened in Panama with the Center for Tropical Forest Studies, 50 hectares, in which every single tree was mapped and identified and followed through time. 50 hectares is a large area, and that is a lot of trees. That has now been done in something like 50 plots in over 20 countries worldwide. 4.5 million trees that have been censused, and many, many partner institutions. It's been an extremely successful program. There have been over 500 publications that have come out of this that have revolutionized the way tropical forest science is done and what we know about it. The reason I bring it up is in part to show that it is possible to do this. This was a big effort aimed at originally at one, as I say, seemingly simple question that actually branched out into a very large number of related questions as a platform for other kinds of research. So what we're interested in doing now is extending that model to the ocean. So through the MarineGEO, beginning with the Tenenbaum Marine Observatories network. This is a network of sites at the Smithsonian, and the coordination that the Smithsonian will have for the network, which was funded by a generous gift by Michael and Suzanne Tenenbaum of the Smithsonian's National Board. And what we're interested in is another deceptively simple question, which is what makes some marine ecosystems resilient and others very vulnerable to environmental change? And we think that the answer to this question has a lot to do with changing biodiversity, as we were talking about earlier. So we are beginning this program, and I'll tell you momentarily about what we're doing and what we plan to do at the Smithsonian's set of field stations in several sites in the Eastern Atlantic and the Pacific. The Smithsonian has a world-class institution at the Smithsonian Environmental Research Center in Maryland. Tuck Hines and Greg Ruiz are here from CERC today, as many of you know. CERC has a number of staff working here at Tiburon and has for some years, primarily on invasive species, but other issues as well. So we have facilities at CERC, at the Smithsonian's Marine Station in Fort Pierce, Florida, Cariboque, Belize, and on both coasts of the Isthmus of Panama. So we're beginning using these sites as sort of a laboratory for working out the methods that will be used in a long-term, global scale study of how environment and biodiversity and ecosystem processes work together. As you will see, I have also put our two premier sites outside the Smithsonian that we're just beginning to develop now, San Francisco State University Tiburon Center here. We're also working at the University of Hawaii on a very similar kind of partnership. We have signed a memo of understanding with both San Francisco State and with University of Hawaii more recently. And so we're looking at these sites as really sort of the pioneers where we figure out how we're going to do it everywhere else. Then there are a very large number of other places where we could expand the network, and I've just put a few of them on here. Most of these are sites where we have had conversations with institutions or individuals there that are interested in joining the network. None of them are certain yet, but the reason I bring this up is to show that there's quite a bit of interest throughout the world in joining this network, and that as San Francisco State and Tiburon begin this process, this will ultimately, we feel quite confident, ultimately come to a very large network of sites which will be very powerful in answering some of these questions that we can't do at individual sites. So, what will we do? The vision, if you will, of MarineGEO might be summarized as a science partnership taking the pulse of the living ocean to keep it healthy. So let me explain what I mean here. So first of all, whoops, I need to go back. Yes. It is about science. That is, the purpose of this effort and the network is to collect rigorous science that we can use to determine how marine ecosystems are responding to environmental change, to conduct experiments to understand why that's happening, and to be able to have comparable standardized data across all the sites that give us a much more powerful picture of how and why things are changing than we have had before. So it's about science. It's obviously about partnership. No institution in the world, including the Smithsonian, would be able to do this alone. Although I will say that one of the reasons why I took this job is I'm convinced that the Smithsonian is the only organization in the world that would be able to coordinate this across such a large scale and large group of diverse partners. And we can talk more about that later if you wish. So a science partnership, taking the pulse of the living ocean. By taking the pulse, we mean keeping track of the vital signs, so to speak, measuring the state of the environment and the state of the organisms that are living there so that we have early warning signals of any potential changes. And then finally, and I should say also, the living ocean is key here, and I'll come back to this in a few moments. And this is because, as many of you know, there are a number of sophisticated ocean observing systems that have spread around the world in recent years, but I think it's fair to say that the vast majority of those are really focusing on the physical movement of water and tracing chemicals through the water and so on. And there's surprisingly little systematic effort to understand the living part of the ocean, the biodiversity, the organisms that are living there in a standardized way. So, and finally, all of that science is ultimately, we hope, directed towards the goal of keeping the system healthy, of understanding how it works so that it can inform policy. So what does that mean specifically? Some of the very general questions that we need to answer, and this will be surprising to some that we don't know the answers to these yet, are what do we have? What biodiversity is out there? So we know this for some places that are very well studied, such as San Francisco Bay. We don't know it for many other sites. And in order to really understand on a global scale how environment is influencing diversity, we need to know, first of all, where it is and who is there. So that we know how to do through intensive biodiversity censuses. Perhaps more important, certainly equally important and of great interest to me is how does it work? What are those organisms doing? Are they just lawn ornaments, so to speak, in the seagrass, or are they actually doing things that are important to us, producing food, maintaining stable shorelines, keeping the system stable? That is really a question for experiments, and I'll show you an example of coordinated experiments in a moment that we already have underway. How is the diversity changing? To really get at this, we need rigorous long-term time series observations, that is monitoring of the organisms and the environment. There is a lot of change from year to year. As you all know, you're experiencing a drought now. At other times we have too much water. We've had a lot of snow in Washington after a number of very hot years. So there's a lot of natural variability. To really get at what the long-term trends are, we need these long time series observations. Why is it changing? Again, we need experiments and modeling. And what can we do about it? So this is sort of as we move outside of the realm of science and into the realm of applied research and policy. And there what we need is ecological restoration projects, which are very active in the Bay by Kathy and others, as we just heard, citizen involvement, and policy action. So these are the very general goals that we have for the partnership. And we're actually meeting with a number of the scientists from Tiburon Center tomorrow to really hash out what the specifics of this plan are and how this can benefit all parties. So I want to take a moment to come back to the issue that I brought up earlier about what is unique about this MarineGEO effort. And the way I would summarize it very succinctly is that we want to know about how biodiversity works. So as I mentioned before, many of this figure, to my mind, really sort of encapsulates what most of the existing ocean observing systems are about. There are a lot of planes and satellites and ships and interesting sensors and machines doing things that are telling us critically important information like how the water moves, where the currents are, when storms are coming, how pollution moves around, how fish larvae move around, those sorts of things. But there's actually relatively little focus here on the animals and plants living in the system. So whereas the focus of the existing systems is primarily physical chemical, our focus is on biodiversity, on the animals and plants that live there and what they're doing. The ocean observing system approach is primarily descriptive and modeling, measuring the water movements and understanding how that's happening. Our approach will also involve experiments, the gold standard, if you will, for understanding the causes and effects of processes in the natural world. Many of the observing systems, although the networks are expanding, the systems are relatively geographically focused, and partly that's because they're extremely expensive. There are a lot of instruments out here. Whereas what we really hope is that by leveraging partnerships with various institutions throughout the world, we'll be able to spread out into a really large network. And finally, the last two I think are really important. There's really no explicit focus on people in most of the ocean observing systems that are happening right now. And this is, to do so is very important. And that is a critical part of what we're looking at, because as we were just talking about, there is a huge impact of humans on the marine environment, and we need to know how we're interacting with the environment, what our impacts are. Part of the way of getting at that is by using history. So by focusing on the organisms, the biodiversity there, and using the Smithsonian's resources, we can go back in time using cores of fossil cores that go down into the sediment and have amazing ability to reconstruct what kinds of fishes were living in the system, how big they were, what people were eating, and so on, back through recent time into the deep past. So this is the niche that we're seeing at this stage is unique about the MarineGEO or Tenenbaum network that is not present in most of the other existing efforts. So what can we do? There's a lot here. I don't want to knock people out right away after the wine that you've had, but I'll just mention a few things to show you that we are thinking about the specifics of what will be measured in the different sites within the network. So we can think of them sort of as the stage, the players, the action, and so on. The stage for all of these biological processes is, of course, set by the environment. So the temperature, all of the water quality, temperature, salinity, the water chemistry, such as pH. The oceans are acidifying, and that has numerous implications for the kinds of organisms that live there. We need to know about that. And there are already numerous efforts underway towards this. We're very interested in leveraging and collaborating with other organizations that are already doing part of this work. In fact, we've been talking with the NERS group here at Tiburon Center, and very keen on adopting their protocols and working with you. On the stage we have the players, which are the organisms that live there, the biodiversity. And so what we intend to do for each of the sites is to find out who's there, that's the taxonomic database, to get molecular data that will give us a much finer scale resolution of the diversity of organisms and how they're related there, as well as image banks, that is photographs of the organisms, food web links, and so on, even seed banks in some cases for organisms that need restoration. And we're working, I'll just take the arrow down from the image bank to this area. I don't believe I have it on there, but we're working with the Encyclopedia of Life to try and use these collections to build a sort of electronic field guide to each site that we work, which will be available to the general public and researchers. The action is what the creatures are doing, how productive is the environment, which of course comes up to harvestable fish, how much of the algae is being grazed, what are predators doing, how fast does carbon decompose versus being stored. This is a very large issue now, as many of you know, is the potential value of coastal vegetation such as marshes and mangroves in storing carbon that's being released into the atmosphere. And then finally, how all of these come together in this box that some of you probably can't see behind the podium of the impacts and services to society: fishery production, shoreline protection, tourism, water quality, the services that I mentioned earlier. So these are in very general outline, are the sorts of things that we would like to develop into a standardized program of research across all of these sites that will make them larger than the sum of the parts. And finally, we'll also be using experiments, as I mentioned, to understand how all of these things fit together. So just to give you a brief sense of what kinds of organisms we're talking about, and I have the coral reef pictures as opposed to San Francisco Bay pictures, partly because you can't get very good pictures of things underwater in San Francisco Bay. And yes, people like to dive on coral reefs, and many of these methods have been worked out there. They won't work everywhere. We have to use some methods differently in estuaries than we do on reefs, obviously. But one of the things that we have in common among all of these different kinds of habitats is habitat-forming organisms. In most areas of the marine environment, as on land, there are certain kinds of plants or even animals that create the habitat for everybody else to live in. In the forests we have trees, obviously the birds and insects, and everybody lives in the trees. In marine environments we have corals, salt marsh grasses, sea grasses, mangroves, oyster beds, and so on. Understanding those habitat-forming organisms is a key part of understanding everything else, because that's what sets the template, so to speak, for everyone else. So there are standardized ways of measuring these and keeping track of them over time. That'll be a big part of this. Can work not only in coral reefs, but in seagrass beds and so on. At the other end of the spectrum from these stationary plants and animals that are creating the habitat are the mobile animals that are living in the habitat. And I single out fishes in particular for a number of reasons. One is, of course, we eat fishes, so it's one of the most direct links from the ecosystem to a service or a product that benefits humans. The other thing is that fishes are what might be called ecosystem engineers. What their grazing and feeding activities have a major impact on who lives in the system and what it looks like. Fishes, for example, can tip the balance between the bottom being covered by beautiful corals and reef versus the bottom being covered by algae that's of less value. Also, the taxonomy of fishes is relatively well known, so we can get started very quickly in cataloging the diversity there. Now, there are many, many kinds of organisms in between these habitat formers on the one hand and the fishes, including a whole variety of tiny invertebrates that nobody really wants to deal with because they're hard to identify. Well, I shouldn't say nobody. Many of us actually do love them and know them. But your average person is generally not so much interested in amphipods and small crustaceans like that. Nevertheless, they are very important in some systems, as we'll see shortly. And there have been a number of sort of ingenious efforts to sample these things. They're very difficult to sample in coral reef environments, for example, because this reef rock is difficult to get at. You have to break it apart to get the animals. They're buried down in there. It's very heterogeneous. So this is what is called an autonomous reef monitoring system, which is sort of a fancy name for a bunch of PVC plates that are all stacked together, and you throw it out on the reef, leave it there for a year or two, and it essentially becomes part of the reef. All of the corals and algae and various different kinds of invertebrates start growing over it and hiding in here because they don't want to be eaten, which is a problem for small animals on reefs. And after the end of a year or two, this can be brought up to the surface, and it has almost all of the organisms that you would find on a reef, except for the large fish, obviously. And so it's a really efficient way of getting a standardized sample of the organisms on the reef. And these are then subjected in a group that includes people at the Smithsonian who developed this assay. It goes through a whole series of methods to get the biodiversity out of it. This is what it looks like after it's been sitting out there. The shape is different, but otherwise it looks like the reef because it's been colonized by everything. And what we get out of these samplers, these standardized samplers, is a series of different ways of measuring biodiversity. So the plates are pulled apart and photographed so you can see what kinds of creatures are growing on them. Here's an oyster of some sort. There are various kind of sponges and sea squirts. Those are then scraped off and saved as specimens. The animals that are larger than 2 millimeters are individually photographed. A voucher specimen is preserved and the DNA sequence is preserved. So we have all of those things linked together. All the stuff that's smaller than 2 millimeters goes into the blender, is blended up and becomes a DNA milkshake that is then, it's possible to barcode that, fingerprint all of the organisms in there simply by doing DNA sequencing. Well, I should not say simply, it's not simple, but it is possible to be done, let's put it that way. Now, this is, I just show this as one example of possible ways of sampling all of these small creatures and microbes and algae and invertebrates that are very difficult to get at in other ways. This is very time-consuming and fairly expensive, but it's actually a whole lot less time-consuming than trying to look at them all under the microscope and identify them. So this is one of the sort of innovative ways that is being used to sample biodiversity of cryptic animals on reefs. We'll have to use different methods for quantitatively sampling the animals that are living in the mud and in the sand in a place like San Francisco Bay, but we're on that too. So those are some of the components of who's there that we are and how we might measure them. And that brings me to what they're doing. And there are a variety of ways of measuring what they're doing that we're experimenting with now. And basically the goal here is to find very simple experiments that can be done in lots of different places and yield data very quickly. To measure important ecosystem processes such as production, how quickly new plant matter is being produced, predation intensity, how much are fish feeding, do they have a big impact on the small animals in the system or not. Similarly with grazing, is the algae being grazed effectively so that corals can grow, and so on. And recruitment, which simply means the rate at which new organisms are coming into the population. And there are ways of measuring that by putting out plates that you collect larvae on. So this is just one example, again from a coral reef environment, of a simple assay that's used to measure the intensity of fish grazing. So some colleagues of mine simply take nylon rope and collect different kinds of seaweeds in the environment, put them in the rope in a standardized way, put them out on the reef, and wait for fish to give them a couple of hours for fish to feed, come back and record what's still left. They do this with a video camera so you can actually see which fish are eating them. Again, that's not going to work in an estuary, but the point is there are simple ways of designing these kinds of experiments that, if done all around the world, can give us a good sense of the relative intensity of ecosystem processes such as how fast food web interactions are happening, how active predators are, how active grazers or herbivorous fishes are, for example. Just one example of how it might be done. And the ultimate goal of that eventually is what I would like to see come out of it are global maps of these ecosystem processes, global maps of diversity, global maps of ecosystem processes, and those coming together give us a global map of ocean resilience. That is, can we figure out why some areas are responding in a very negative way to fertilizer dumping and fishing and so on, whereas other ones seem to be okay. This is what I think we can get out of this through a collaborative project such as this. And speaking of collaborative projects, I want to spend just a couple of minutes to show you how this might happen in the marine environment using the example of the seagrass network that Kathy mentioned. Working in eelgrass, which occurs here in the San Francisco Bay and all over the northern hemisphere, actually. The scientific name of eelgrass is Zostera marina. And so what we've done is to gather a group of collaborators from all around the northern hemisphere in what we call the Zostera Experimental Network, or ZEN. This is our cool logo that we put on t-shirts. And I'm not going to spend a lot of time on this because I know it's late, but I'll just say that what we've done is to find collaborators that work in these eelgrass ecosystems all over the northern hemisphere. that were willing to work with us in much the same way as we're talking about with our colleagues here at Tiburon, to do some very simple experiments that become extremely informative when we look at them across sites. And without going into the details, I'll just tell you that we had a whole bunch of sites, 16 actually in the first generation. We're now moving into the second generation, including San Francisco Bay site that was headed up by Kathy Boyer. And basically the experiment consisted of artificially fertilizing seagrass plots out in the field and also taking the grazers away using a degradable insecticide. Very simple, but we looked at how the results of that experiment varied across all of these different sites along gradients of biodiversity, temperature, latitude, and so on. And just to give you one little tidbit of what happened out of it, one of the things that came out that I thought was quite interesting is that we found a pretty strong correlation between diversity and the ability of animals, to the grazers, to keep the grass clean. These seagrass habitats are very important nurseries for fishes and shellfish, but they're very vulnerable to being fouled by algae that overgrow them and cause the death of the seagrass. So these small animals, the bugs and slugs that are out in these systems, despite being very inconspicuous, can be extremely important to the health of the seagrass. They can also be pests on the seagrass, as they are, as some of them are here, some of the invaders are here in San Francisco Bay. But in essence, when we looked across this big network, and these data look pretty noisy, I'll admit, but if you trust me for a moment, what we found is that the sites that had many grazer species, that had high biodiversity, were actually places where we had really effective grazing of that algae and removal of the competitors that maintained healthy seagrass. So that was a really interesting result that sort of resonates with this hypothesis that biodiversity can provide resilience. So that's something, the kind of thing that we're hoping we can get a much better handle on as we go forward with the larger network. And I'm delighted to say that the Zen network has been funded for a second generation by the National Science Foundation, and we're actually expanding out now to 25 different partner institutions in 14 countries and about 50 sites. And one of my secret plan is to ultimately have those sites come into the MarineGEO network as well. And we have a few takers on that so far. So I think that there's a lot to be gained there. Okay, so where do you fit in? And this was addressed primarily to the scientists here at the Romberg Tiburon Center, but of course I would open this to everyone because we need help from all quarters. And these are just a couple of the reasons why we think that joining into this partnership will be valuable to both parties. So by developing this global network of sites, we have a platform for comparative research with environmental data, biodiversity data being collected in the same way at all these places. It creates a real wealth of additional data that we can use to ask questions that are of interest here for San Francisco Bay, as well as for the world at large. Long-term environmental and biological data, there are relatively few places where we can compare those long-term trends over large areas. Explicit connections to other sites worldwide, developing networks with other scientists. This is, we think, especially important for students, and we're talking tomorrow in more detail about how to involve students in this process. We think it's really a win-win situation to have students working on the undergraduates and graduate students as well, working on this research and gaining insights from it. And that sort of segues into the research support and training. We have just recently awarded 2 postdoctoral fellowships, and in fact both of them in the MarineGEO, the Tenenbaum Marine Observatory Network, both of those fellowships are going to recent PhDs that have been working in the San Francisco Bay Area. So we're integrating them into the global network. In fact, Lisa Shealy is here right now. And we're also hoping ultimately, yes, let's have a hand for her. And I should also call out again our gratitude to the Tenenbaums, Michael and Suzanne Tenenbaum of the Smithsonian National Board, who have provided the initial funding for this to get the network off the ground, and who are also supporting that fellowship. There are lots of other opportunities if you'd like to speak with me about that. So research support and training, we think this will actually be quite critical in engaging new talent, in building the network over time. Small grants for research are something that we're looking forward to in the future. Opportunities for exchange not only of students but of scientists and developing a network. And then finally, the economy of scale. Large research questions, increasing the ability of all of these parties to be competitive for grants from agencies and elsewhere. And of course, just in general, collaborative analysis and synthesis. I think this is a revolutionary way to do science. It's the way that science is going, and I very much look forward to San Francisco State being a part of that and to working with you. And I think that might be the end. What all do we gain? I've talked about some of this already. The critical data on nearshore ecosystems, the collections, physical specimens, genomic data, and images, all of which ultimately will be open access, not just to the partners, but ultimately to the general public. Training the next generation, I've talked about already, and partnership in the world-class science. So thank you very much for coming and for your attention, and I would be happy to answer questions. I'll stay here.
All right, now is your chance to participate in the discussion. So please, if you've written down a question, send it over to the end of your row and we'll collect them. If you have questions about the topics that Emmett covered tonight, if you have questions about the Romberg Tiburon Center, about the Smithsonian, any of those are fair game. So please let us know what you're interested in and we'll get those questions up to Emmett.
So the first one is what kinds of global questions can be answered by having monitoring sites around the world? I think by having these sites, and I would say especially by having sites that are both monitoring and experimental, coordinated experiments, we can answer questions that are really impossible to get at by research at individual sites. And that's certainly, from my own perspective, something that I've learned from the Zen network. One of the ways that I look at it is over the course of the last 50 years of marine ecology, the low-hanging fruit has been picked, so to speak. We have a lot of insights about how species interact with one another, what effect predators have on diversity, how nutrient pollution influences algal growth, and so on, at very small scales. But most of the really big and really thorny, challenging questions we now face in the environment are about how large-scale processes interact with those small-scale processes. And these are things like how biogeography and dispersal across very large areas influence how these systems work and how they respond to problems. And that's something that I think we can only get through these kinds of large-scale partnerships.
I can't read all the writing, so that's why we're back here trying to figure out what some of these say.
Do I get to pick the one I like? Or?
Yeah, the one you can read? How about that? All right, let's see. What methods, if any, will be used to assess biodiversity
of pelagic organisms?
Such as nekton and plankton?
Okay, very good question. What methods, if any, will be used to assess biodiversity of pelagic organisms such as nekton and plankton? Obviously, this was written by a scientist. So, pelagic organisms, meaning in the water. Most of the things I've talked about are on the bottom. The nekton and plankton, of course, are the small creatures that are floating around in the water that form the base of the food web. And the nekton are swimming things, which are mostly fish, but also squids and some crabs. So the question, I think, is since we've talked all mostly about the ecosystems on the bottom, what about what's in the water, which obviously is a big part of what's going on, particularly as we get out into deeper water where most of the habitat is in fact the open water. And I guess a couple of things I would say to that. We are very much interested in keeping track of the plankton because that is more or less the food for most of the organisms in these systems, as well as algae that are growing on the bottom. That's critical to know about, and it's very variable through space and time. So I haven't mentioned that, but we're certainly interested in monitoring plankton. As far as the nekton, the fishes, I've already mentioned those. Mostly in association with the fishes that are living near the bottom, associated with seagrass and reefs and so on. Our focus, one of the things that is perhaps not unique, but I think special about this network that differs from many of the other sort of organized long-term observing systems is we're focused on very shallow water, very near shore water, starting actually on land and moving into the water. And the reason for that is that that's where people interact with the ocean for the most part. That's where most of the diversity is, and that's where most of the productivity is. And so in answer to the question, we're primarily interested in the fishes and the plankton that's in shallow water that is interacting with these systems on the bottom, primarily.
Okay. Are you considering, how do you think contaminants should be considered?
Are you considering, or how do you think contaminants should be consolidated in your network site comparison? I think they should be. You know, one of the challenges we will be facing, we are facing now, is what is of the very large world of things that we would like to do, what is realistic to do? And so my feeling is we start with some of the basic things that we've talked about here, and that as that gets underway, then we can expand. Contaminants, pollutants is definitely something that's of keen interest. And in fact, I was just advised by one of our Smithsonian staff who goes and talks to people in Congress and in the agencies that it would be a good idea to monitor pollutants. So we may well be trying to do that soon. Part of the issue with that is that it takes generally fairly specialized chemical expertise that is a little bit different than some of the other things that we're talking about. Although, of course, the molecular analysis of the biodiversity is also sophisticated too. So yes, we would like to do that, and I'd love to talk to any of you who have suggestions about that.
Okay. How will MarineGEO adapt to address the vision need for ecological restoration, citizen involvement, and policy change? It's a bunch of questions all.
Okay, yes, it's a multi-question. How will the program attempt to address the need for ecological restoration, citizen involvement, and policy change? And there's another one here. Let's just do that one for now. Well, these are all things that we have talked about at length and that are in our original white paper that was the sort of dream, the vision for MarineGEO. Where we are now, I think, is in trying to set sort of a series of priorities. Meaning, here's what we'll start with, as I just mentioned, and here's where we go next. We're very keen, as I said, on education generally and on having citizen involvement as well, particularly where we can match citizen involvement with the kinds of research that we're doing that needs a lot of people and that is relatively straightforward measurements that are also very meaningful. And we've seen a number of cases where this can be extremely powerful. You perhaps know about the Christmas bird count that's done by all over the world, I think certainly all over the country. There's a huge quantity of information on bird diversity and abundance and how it's changed over the years that's collected by citizen bird watchers. It would be really neat to do that with the marine systems. There are actually some there are some programs in Australia I know of, and perhaps here as well, that use iPhone apps to try and report new findings of invading fish species, for example. Maybe there's something similar here. So I think as a group, we're at the Smithsonian, certainly, we're very interested in finding ways to do that. We haven't got into the details yet, partly because we're starting with the the basic science architecture, but very interested in that, in bringing in citizen science. Policy change, that's sort of a further downstream one, but I know that a number of us, both at Tiburon and at the Smithsonian, are interacting on a regular basis with managers and policymakers, and so we hope that will continue, and that this data will really provide some powerful information to take there.
Okay, well, that's a good segue into this. So 2 of these have to do with political stuff.
Mm-hmm.
I don't know what this word is. Does the Smithsonian have a plan for political something? And then the second one is global political interest in Timon as strong as global scientific interest.
Okay, that's interesting. Does Smithsonian have a plan for political something? I don't even know what the last word is, but I can answer it. And the answer is no. We do not have a plan for political anything. The Smithsonian is Switzerland, is the way it was stated to me, and I really like that. The Smithsonian could be described as a public-private partnership. Some of our money comes through the federal government. Some of it comes from endowments and so on. But we are. Can I say this? Is a federal agency? No, sorry. I wasn't supposed to say that. At any rate, the point is we are an apolitical entity, and our job is to provide reliable scientific information and other kinds of information. And I want to stress how important that is. We, like any reputable scientific organization, rely on people's trust that we're objective. And so we cannot be political, and I think I would also sort of come even back a little bit further from that to the question of whether we can be activists, so to speak, and maybe that's the wrong word. The way I would say this is we are happy to provide, and in fact dedicated to providing, objective scientific information that is of use to making policy decisions. But I think the position we have to take is say, this is the science as we understand it. If a policy has been recommended, I would feel comfortable saying, to the best of our scientific knowledge, this is what would happen if this policy is enacted. This is what would happen if it is not, to the best of what we understand now. But we cannot advocate for a particular policy. My boss, Tuck Hines, might say something about that. I don't know, but that's correct. Okay, good. I got that right. Let's see. What was the other one? Political. Is global political interest in Timon as strong as global scientific interest? I'm not sure. I'm not sure I know actually what that means. Most of the contacts that we've had with potential partners outside the U.S. have been mostly with individual scientists or with administrators of organizations, of academic institutions, and there is pretty strong interest in a number of places there. I am quite optimistic about the ability to get started in those places, because in most of them my sense is that there would be a strong interest in partnering with the Smithsonian. There will of course be some places that would be difficult to work for various reasons, both political, safety, and logistics and so on. So those are things obviously that we'll need to take into consideration as we develop the plan.
I'm getting low on questions, so if there's things that are building in your brains, we have more cards. Please raise your hand and we'll pass them out to you. Otherwise we'll end sooner than we thought we would. How do estuaries make clean water? Isn't it salty?
That's a good question. How do estuaries make clean water? Isn't it salty? Well, it is in fact salty. In some places more than others. And this shows me that I wasn't being clear. When I say clean water, I mean water that is relatively free of sediment and debris and pollutants. What I mean by estuaries making clean water is that not the estuary per se, but the vegetated habitats along the margins of the estuaries. Because as water comes in on the high tide and goes out on the low tide and pools in those areas, first of all, the vegetation there slows it down, and that allows sediment and mud and sand particles to fall out onto the bottom. Also, there are many filter-feeding organisms in there that are eating that stuff up, and some of them are also concentrating the pollutants either in their own bodies or down into the sediment, and so that when the water eventually washes out again, it's relatively cleaner in the sense that it doesn't have a lot of mud in it. Sometimes the phytoplankton, the algae in the water has been eaten by filter feeders. That's what I mean by clean. It's still salty. I wouldn't drink it. Yeah.
Okay. People who deny global climate change seem to be in positions of political power and influence. What can be done to counter the harm these people cause?
All right, I'm going to read this question. It's not my question. People who deny climate change seem to be in positions of political power and influence. What can be done to counter the harm these people cause? I just answered the question a few minutes ago that I'm not getting political. So, it's probably best if I just don't even touch that. But what I will say is that part of the reason that there's argument about climate change is because the data are inherently noisy. It's understandable. This is a very complex problem. I personally am quite convinced by the data that it's happening and that we have problems coming. But I can understand that this is a difficult issue. This is one reason why I think the kinds of things that we're talking about here are critically important, because the only way to really show this is to have good data collected from a variety of different places where we can really where we have really objective data that shows what's happening, and we really only have those kind of records on a long term from a couple of places at this stage.
That's all the questions we have.
Thank you.